Hello and welcome to this video about the COVID-19 vaccines. For this video I would like to provide some information about how the COVID-19 vaccines work and give a brief scientific explanation. I would also like to take some fear from those of you who might have doubts about the vaccine and actually encourage you to get vaccinated as soon as you can. If you have any questions about the content, please post them in the comments down below and I will answer them as soon as possible. If you would like to listen to the German version of the video, please check the video description where I post a link to the different version. The outline is to answer a few questions. First of all, how does a vaccine work in general? How are proteins produced inside a cell, which is going to be crucial to understand how the vaccines work? How does an RNA vaccine work? How does a vector vaccine work? And so basically, what's the difference between these two types of vaccines? And how effective is the COVID-19 vaccine against variants which have been spreading throughout the globe? So let's talk about vaccination in general first. Here we distinguish between active and passive immunization. An active immunization has a long-term effect, whereas a passive immunization has more of a short-term effect. In any case, we want to fight a pathogen. A pathogen can be a virus or bacterium, basically anything that causes a disease to our body, but we can train our immune system with a harmless version of this certain pathogen. So our immune cells can produce antibodies, and these antibodies bind to the pathogen and prevent it from spreading throughout our body. But to figure out which antibodies are the right ones, our cells have to check if they can make the right antibodies. So they will interact with the pathogen and figure out, oh, this is not the right one. The next one can come in, and also this one doesn't fit perfectly. But eventually, one of the cells will have the right type of antibodies to bind to the certain pathogen, and then get a positive feedback signal. From this, it will then replicate and make more of itself. These new cells can produce a lot of antibodies, which can also bind to the pathogen and basically tag it for being degraded, but also stop it from spreading throughout our body. However, over time, the antibodies and these cells will degrade and vanish from our body, but a small subpopulation of these cells are so-called memory cells. These memory cells will last over a long time in our body, and they can facilitate a quick immune response upon a repeated infection. The passive immunization, on the other hand, is a little bit different. Also here, in this case, our body has to deal with a certain pathogen. But in this case, we only administer antibodies straight into our body. These antibodies can again then tag the pathogen for being degraded and stop it from spreading throughout our body. But again, the antibodies will vanish over time, so the passive immunization is more of a booster to help our immune system just in this particular moment, but not on the long term. So no memory cells are being formed, and when the pathogen re-enters the body later on in our life, our immune response is still slow, in contrast to the active immunization where the memory cells can respond quicker to repeated infections. So next, let's talk about protein biosynthesis. This is an important principle that we will re-encounter on the next few slides. Our body is made up of many cells, and each cell contains a cell nucleus, which stores our DNA and protects it. However, we need the DNA to make proteins. For example, gene X will code for protein X. But we cannot just take gene X out of the cell nucleus. Instead, we can transcribe this gene into a messenger RNA. So for gene X, it would be mRNA X. This mRNA can then leave the cell nucleus through nuclear pores. After the mRNA left the cell nucleus, it cannot get back into the cell nucleus. So these nuclear pores are one-way streets for mRNA. Once the mRNA is in the main part of the cell, so-called ribosomes can read and translate the information on the mRNA to make the protein X. Protein X can then be used for whatever its purpose is. This is an important principle, like I said, that we will re-encounter in parts on the next few slides. But usually we always need DNA, transcribe it into RNA, and the RNA will be translated into a protein. So let's talk about the COVID-19 vaccines now. First of all, the RNA vaccine made, for example, by BioNTech or Moderna. These utilize a technique where a vesicle will deliver the spike protein mRNA to your body cells. The spike protein is a protein that's coating the surface of the virus, and this spike protein can be recognized by antibodies from the immune system, which make them realize this is a pathogen, this is harming our body. So once the vesicle fused to our cell, the mRNA can be released into the main body of the cell. From here, the mRNA cannot enter the cell nucleus, so our own DNA is protected and the virus DNA, the virus information, cannot be integrated into our genome. So remembering, usually we go from DNA to RNA. Basically skipped the step of the transcription here already, since ready-made mRNA was delivered to a body cell. Now, ribosomes can again read and translate the information on the mRNA and make a protein, the so-called spike protein. The spike protein can then be brought to the surface of a body cell and be displayed to immune cells. So now immune cells can train with this spike protein and test if they can make the right antibodies, like we saw earlier. This one doesn't fit. Next one comes in. Oh yeah, this one fits. And now we'll make more of itself. Part of these cells will be antibody producing cells, making a ton of antibodies that can also bind to spike protein. Another subpopulation of these new cells are the memory cells, which will be helpful if the actual COVID-19 virus will enter our body to have a quicker immune response and fight the disease right away. Next, let's talk about the vaccine made by AstraZeneca by Johnson & Johnson. It works similarly, but still a little bit different. And the main difference is by delivery of the mRNA. These companies use a disarmed flu virus, so basically only the shell of the virus, which only contains the spike protein mRNA again. This disarmed flu virus can then inject the mRNA into our body cells, and once the mRNA enters the body cell, again, it cannot enter the cell nucleus, and our DNA is protected from any viral DNA. Again, we skip the step of transcription from DNA to RNA, and we get ready-made mRNA delivered into our cells. Our cells can then use the ribosomes again to translate the mRNA into the spike protein, and the spike protein can be displayed on the cell surface for our immune system to train. So it's kind of like a dummy again. Different body cells can come in and test the antibodies if they would fit the spike protein, until eventually one of them figures out, yep, I got the right antibody and I will replicate a lot now. Part of these cells again are antibody producing cells. They can tag the spike protein on the cell surface of our cells, but also on the virus. And part of them are the memory cells that will have a long-lasting effect and help upon a serious infection with the COVID-19 virus. So now let's talk about how the immune response works upon a COVID-19 infection after we get vaccinated. Successful vaccination will lead to memory cells being distributed in our body, and these memory cells can fight the COVID-19 virus faster. They will bind to the spike protein on the surface of the virus and immediately realize this is a pathogen, I have to fight it. Therefore, they make a ton of antibodies, and these antibodies can bind to the virus, tag it for degradation, and stop it from spreading. So memory cells will produce a quick, specific antibody response to the virus particles and stop them from spreading through our body before they can actually harm our body cells, before more virus particles are being formed. So now let's talk about how the COVID-19 variants might affect the efficiency of the vaccine. Here we have three different scenarios. The first one is a so-called silent mutation of the spike protein. That means, although there's been a slight change to the protein, the overall structure hasn't changed. And this means that antibodies, which have previously been proven effective against the spike protein, can still bind to the virus and stop the spread throughout our body. In this case, the vaccine will still be effective. A different scenario is a mutation of a different protein other than the spike protein. In this case, of course, the spike protein hasn't been changed at all, and the antibodies can still bind to it, stop the virus, and the vaccine is also still effective. The last case is a structural mutation of the spike protein. Here you see the triangle shape has been changed to a dome-like shape, and Now the structure is changed so that the antibodies cannot bind and detect the virus, and therefore they cannot stop the spread of the virus. In this case, the vaccine will be ineffective. However, this has a low probability. The spike protein is also important for the virus to actually infect our body cells. And if there's been a spike protein mutation that changes the overall structure, it might also be the case that the virus cannot actually harm our body cells anymore. So let's recap the content of this video. How does the vaccine work in general? Antibodies, antibody-producing memory cells are formed, and especially the latter are very helpful upon a reinfection with the same pathogen. How are proteins produced inside a cell? Transcription of DNA into RNA is followed by the translation of RNA into protein. How does an RNA vaccine work? RNA gets delivered by vesicles into cells and is translated into spike protein. The spike protein can then function as a training dummy for our immune system. How does the vector vaccine work? RNA gets delivered by a harmless virus into cells and is translated into spike protein. The spike protein again can help our immune system train against a severe virus itself. How effective is the COVID-19 vaccine against variants? Well, only severe mutations that render the vaccine ineffective will actually cause trouble. So thank you for watching the video. I hope you learned something. If you have any questions, put them down in the comments, and stay safe, get vaccinated, and don't forget to wear a mask.